Hi everyone, good morning to all. I am Mrs. Kokila. I'm going to talk about enzymes. In this video, we are going to see about what is an enzyme nomenclature, characteristic features of enzymes, enzyme structure, and enzyme classification. Let us see enzyme. What is an enzyme? Enzymes are biological catalysts that speed up the rate of biochemical reaction. Most of the enzymes are protein in nature, except ribozyme, which is an RNA acting as an enzyme. Enzymes, they act on substrate and convert it into products. I've shown a figure down which indicates that the substrate binds to the active site of the enzyme and forms a complex called as enzyme substrate complex. It is an intermediary complex which undergo reaction to produce products A and B, and enzyme is released. What is an enzyme and how it is named? The term enzyme was coined in the year 1878 by F.W. Kone. The name enzyme was derived from a Greek word en meaning in and zyme meaning yeast, which literally means in yeast. Enzymes are generally named according to the reaction they catalyze and by suffixing ASE after the name of the substrate. Later, the International Union of Biochemistry developed a standard nomenclature for enzymes. Each enzyme is described by a sequence of 4 numbers preceded by EC. EC, it denotes enzyme commission, and the number of enzyme is called EC number. When classified, each enzyme is assigned the EC number in the form of digits separated by periods. So in this, the first number categorizes the enzyme based on its reaction. The first 3 digits of this EC number represent the class, subclass and sub-subclass to which an enzyme belongs to. And the fourth digit is a serial number to identify the particular enzyme within a sub-subclass. The class, subclass and sub-subclass provide additional information about the reaction that is catalyzed by the enzyme. For example, in case of the given EC number, EC 1.2.3.4, the digits indicate that the enzyme is an oxidoreductase, which is denoted by the class 1 enzymes. And it acts on aldehyde or oxo group of donors. So the second digit, which indicates subclass, is given 2. And since oxygen is an acceptor, sub-subclass becomes 3. And that it was the fourth enzyme classified in this sub-subclass. That's why the fourth digit is given the number 4. Now let us move to the characteristic features of enzymes. How the enzymes act and what are the important features of the enzymes. Enzymes are highly specific to the reaction they catalyze. Not only to the reaction but also to the substrate, it is highly specific. The enzyme accelerates the rate of the chemical reactions that occur in a cell by lowering the activation energy of the reaction. The enzyme remains unchanged after a chemical reaction. They are sensitive to various factors which includes pH, temperature and substrate concentration. Even the presence of any inhibitors or activators also influence the rate of the reaction. The enzymes catalyze reversible reactions also, and in certain cases, even small amount of enzyme is sufficient enough to speed up the chemical reactions. Now let us move to the structure of enzyme. The enzymes are composed of structure, apoenzyme, which is the protein part of the enzyme. So in the figure which is depicted here, it indicates that the apoenzyme is the protein part of the enzyme. When this apoenzyme is bound with the cofactors or prosthetic groups, then it becomes hollow enzyme. Apoenzyme, it is the protein part of the enzyme, whereas hollow enzyme, it represents the apoenzyme bound to the cofactor or prosthetic group. Now, what is an active site? The active site of an enzyme is the region that binds substrates, cofactors, and prosthetic group and contains a residue that helps to hold the substrate. Actually, this active site occupies very less portion, that is less than 5% of the total surface area of the enzyme. And this active site is highly specific in shape due to the tertiary structure of protein. Upon denaturation, this active site will lose its activity because it loses its tertiary structure. And these active sites consist of two important regions. One is catalytic site and binding site. Catalytic site is the portion where the reaction occurs, whereas binding site is the portion where the substrate or other cofactors bind to the enzyme. Coming to cofactors, what are cofactors? Cofactors are non-protein molecules that carries out chemical reaction and are required for proper activity of the enzyme. These cofactors can be categorized as organic cofactors and inorganic cofactors. Organic cofactors, for example, PLP, NAD belongs to organic cofactors, and certain cofactors like metal ions, zinc, magnesium, they categorize under inorganic cofactors. These organic cofactors in turn are classified into prosthetic group and coenzyme. Prosthetic group are those cofactors which are tightly bound to the enzyme. So these are tightly bound organic cofactors, for example, flavin and biotin, whereas coenzymes are loosely bound organic cofactors, for example, NAD+. Coenzymes. When you talk about coenzyme, it is interesting to see its features, like it is a low molecular weight organic substance and without which the enzyme cannot exhibit any reaction. And they are actually loosely bound cofactors. These coenzymes are directly involved and altered in the course of chemical reactions, and those cofactors are considered to be a type of secondary substrate. So we call coenzymes as secondary substrate sometimes because it becomes altered in the course of chemical reaction. Many coenzymes are closely related to vitamins. These coenzymes are involved in transfer of groups like hydrogen and various other groups, thereby it brings about a reaction. For example, ATP, FAD, NAD, they are some of the examples of coenzymes. In this slide, you can see different types of coenzymes and from where they are derived and what type of reaction they participate in. First, for example, TPP, thiamine pyrophosphate, which is a coenzyme derived from vitamin B1 or thiamine, and it is involved in oxidative decarboxylation. The coenzyme FAD is derived from vitamin B2 or riboflavin, and it takes part in oxidation reduction reaction. Coenzyme A, which is derived from vitamin B3 pantothenic acid, it is involved in acyl group transfer. PLP is a coenzyme derived from pyridoxine, that is vitamin B6, and it is involved in transfer of groups to and from amino acids. 5-deoxyl-adenosyl-cobalamin, which is a coenzyme derived from vitamin B12, is involved in intramolecular rearrangements. The coenzyme NAD+ is derived from niacin and it takes part in oxidation reduction reaction. Tetrahydrofolate is a coenzyme which is derived from folic acid and it takes part in one carbon group transfer. Biotin, which is a coenzyme, it involved in carboxylation reaction. So these are some of the coenzymes and the reaction type which they participate in. Now let's move to classification of enzymes. The enzymes are classified in many ways. One such classification is enzymes which are classified based on the reaction they catalyze. Based on the reaction they catalyze, the enzymes are classified into 6 major classes. Class 1, 2, 3, 4, 5 and 6. First one is oxidoreductases. These are group of enzymes which catalyze redox reaction, and it can be categorized into oxidases and reductases. For example, alcohol dehydrogenase. The second major class are transferases. Transferases are enzymes which can catalyze the transfer or exchange of certain groups among the substrates. For example, hexokinase. Third class is hydrolases. These are enzymes which catalyze the hydrolysis of their substrate by adding constituents of water across the bond they split. For example, trypsin. Fourth class of enzymes are lyases, which promote the removal of a group from the substrate to leave a bond, double bond reaction, or it catalyzes its reverse reaction. Aldolases are one of the lyases. Fifth classes are isomerases. These are enzymes which facilitate the conversion of isomers, geometric isomers or optical isomers. For example, glucose phosphate isomerase. Sixth classes are ligases. Ligases are enzymes which catalyze the synthesis of two molecular substrates into one molecular compound with energy. For example, glutamine synthetase. So this is how the enzymes are classified into six major classes based on the reaction they catalyze. In a nutshell, we have learned today about the enzymes and nomenclature, structure of enzymes, coenzymes and cofactors, and how the enzymes are classified. Hope you understood. Thank you.